double helix. All right, so let's talk about DNA structure. So DNA is a double helix. The backbone of DNA are the sugars and phosphates. So if you look at just this thing right here, so look at my cursor, you have a phosphate, a sugar, and a nitrogenous base. And these are the 4 different options for nitrogenous bases. So these are the 4 different types of nucleotides. They are bonded together here. So you can see the sugar and the phosphate. So these are 4 nucleotides bonded together, making up a strand of DNA. And so the backbone of DNA is the phosphate and sugar. And it's kind of like the rungs, like if this was a ladder, because you have 2 of them. And I have a really good picture later on. The rungs of a ladder. So if you have a backbone over here, and then you have the complementary strand on the right side, you'd have the backbone over there, and then you'd have the base pairing occurring in the middle. The nitrogenous bases, like I said before, are adenine, guanine, cytosine, and thymine. Don't write this down. You don't need to do purine and pyrimidine. I don't care if anyone knows that. Even in AP, I don't care if you know that. There's like, okay, adenine and guanine are double ringed and thymine and cytosine are single ringed. Like, who cares? I'm sure somebody does, but not for this class. So adenine and thymine are going to base pair. Guanine and cytosine are going to base pair. Once again, I had said that earlier under Chargaff's rule, but this is just giving you the picture for it. Okay, so here's a really good picture of DNA. This picture right here doesn't give you the complete view of what DNA actually looks like. This one gives you a good view of what DNA actually looks like. So here's A is going to show you key features of the DNA structure, very simplistic model. B is going to show you kind of the chemical structure. And then C is going to show you the space filling model, it doesn't give any letters or anything like that. It's just kind of showing you the clouds, like the atoms and their clouds and stuff like that. So here's what I need you to know. I need you to know, kind of like what I said before, that one nucleotide over here is going to base pair with another nucleotide over here. These are the 2 strands of DNA. And that between 2 nitrogenous bases, you have what's called a hydrogen bond. And they're going to hold the molecule together like a zipper. Hydrogen bonds are strong attractions, but they're weak bonds. That's why they're represented by a dotted line, because they're that weak. There really are just a strong attraction between a partial positive and a partial negative charge between 2 different molecules. But I need to write down they're held together by hydrogen bonds, and the shape of DNA is a double helix. And so what causes the double helix shape? What causes the double helix shape is the anti-parallel nature of the 2 strands. So what I'd like for you to write down is the 2 strands run counter to each other. That's what anti-parallel means. They run counter to each other. One strand, the phosphate end is up on the nucleotide. On the other strand, the phosphate group is down. So these nucleotides are turned upside down. And then I'd also like for you to write down the 5' end. 5' just refers to, if you look at my cursor here, you see where the oxygen is? If you count the carbons to the right of the oxygen, 1, 2, 3, 4, 5, the phosphate group is hanging off of the 5th carbon. And then it's the same thing here. If you look at the hydroxyl group, the OH, it's 1, 2, 3. It's hanging off of the 3rd carbon. So you're going backwards since that's upside down. So the phosphate group is on the 5' end, the hydroxyl group is on the 3' end, and anti-parallel means that the 2 strands are running counter to each other, and because of the negative charges on the phosphates, it's going to cause a wrapping. So the anti-parallel running counter to each other is going to cause a wrapping, and that's what the double helix comes from. Okay, DNA is, and this is in eukaryotes, so write eukaryote on top of here, right? Eukaryote. In a eukaryote, DNA is wrapped around proteins called histones. and that's how it's packaged. And then I'd also like for you to write the word chromatin at the bottom. Chromatin. And I've mentioned that when I talked about the nucleus the other day. So DNA is wrapped around these proteins and it's packaged in DNA with its associated proteins, and those proteins are called histones. It's called chromatin, right? And eukaryotes have that. Chromatin, right before a cell divides, is going to further condense into chromosomes. And you don't need to write anything down about this slide right here. The only thing I want to say is that I'll just talk about that a little bit more when we do mitosis and cell division. All right, let's compare DNA between prokaryotes and eukaryotes because there is a big difference. Remember, prokaryotes are your bacteria that don't have a nucleus. They're very simple, no membrane-bound organelles. Eukaryotes do have a nucleus and do have membrane-bound organelles. The one thing they do have in common is that DNA is double-stranded in both. You wouldn't believe how many times students will tell me prokaryotes have single-stranded DNA. No. And I think they misinterpreted what the question actually was, and I'll tell you in a second what I think they thought. DNA is double-stranded no matter what. It's always double-stranded. In prokaryotes, DNA comes in a circle. The chromosome is in a circle. And in eukaryotes, the chromosome is actually linear. In prokaryotes, they only have one chromosome, and that's what makes it circular. There's just one circular chromosome. Eukaryotes are usually going to have more than one, so more than one chromosome in eukaryotes. The DNA of prokaryotes is found in the cytoplasm, and in eukaryotes it's found in the nucleus. And then eukaryotes are going to have DNA wrapped around histones. Prokaryotes have no histones, and DNA with its proteins is called chromatin. And then in prokaryotes, it's just supercoiled DNA, just DNA by itself. So if you need to keep writing, go ahead and pause because I'm going to move on. It's important to know those differences. Let's talk about DNA replication. So DNA needs to be copied before a cell divides. It needs to be copied because the new cell needs the same DNA in it, right? So we are going to go through how DNA gets copied. Cross out the question that says who proved the semi-conservative model. It was the guys called Messelson. I don't even remember the first guy's name. I feel bad. Messelson and Stahl, and they just proved this what's called the semi-conservative model. So you can cross their names out. I'm not going to ask you their names. Or you can cross the question out because I'm not going to ask you their names. But it's important to know what semi-conservative means. So on A, your parent molecule, that's also known as your original. So I would write original underneath that. So your original DNA. And as you can see, one strand is complementary to the other strand. A goes with T, C goes with G, T goes with A, T, C, G. So if I gave you this left side right here, you should be able to tell me what is going to be on the right side. Okay, so this is your original DNA. It gets separated, and each new, the cell is dividing, and so each new daughter cell gets one strand that was from the original and one new. One strand that's from the original and one new. And so that's what's called semi-conservative, is that the daughter DNA molecules each consist of one of the parent strands and one of the new strands. DNA